In this video, we will discuss about the role of Muc-1 in the breast cancer. So Muc-1 is the protein that are Muc-1 is the protein, glycosylated protein that is expressed on the cell surface that can be seen here, and it has both the cytoplasmic domain and also the extracellular or N-terminal domain. And in the case of breast cancer, this Muc-1 is overexpressed on the cell surface of the breast tumors, and because of that it will leads to the activation of several downstream signal. The Muc-1 cytoplasmic domain can affect the intracellular function by manipulating several by manipulating the normal functions of the cells. For example, it can activate this TGF beta related kinases, and that might leads to the survival or metastasis, angiogenesis or proliferations of the cells. That might trigger, you can say that, uncontrolled growth rate and metastasis, that is spread of these tumors from one part of the body to other part. At the same time, it inhibit the function of p53. P53 is the proteins that plays a role to determine whether the DNA damage within the cells is repairable or not. If it is repairable, it can prevent the apoptosis, or if the DNA damage within the tumor cells is not repairable, then it might trigger to apoptosis. However, since the p53 has been inhibited by Muc-1, so the cells will not go for apoptosis through these p53 pathways. The other ways, the other things that might affect is through this P13K AKT pathway, and that will lead to this hypoxia alpha proliferation that we have discussed in one of our previous videos, how this HIF complex plays a role in proliferation or metastasis of the tumor cells. It will interact it will interact with the uh, promoters of the genes that are responsible for the survival and proliferation of the breast cancers. And this STAT3, the role of STAT3 is similar to that of this NF kappa beta pathway. That is, it helps in the survival, metastasis, and angiogenesis and proliferation. So these figures has been shown in the zoom in view, or in slightly in-depth information on the right hand side. In this case, we can see that how this Muc-1 cytoplasmic domain interacts, that might lead to triggering of this pathways that are shown here. So this Muc-1C, when Muc-1 gets activated, or N terminal of domain of Muc-1 gets activated, it will lead to the phosphorylation of this Muc-1 cytoplasmic domain, and that phosphorylation leads to the complex formation of this SH2-GRB2, and this will trigger SOS, and this uh, RAS pathway will then get activated, and thereafter it will lead to this RAF and this MEK uh, 1,2 kinase pathway, and this will lead to the proliferation and survivals, upregulations of the genes that are involved in proliferations and survivals. The other thing is that this Muc-1 interact with this GSK3 beta, and this complex then interacts with this beta catenin, and this will lead to upregulations of the factors that are involved in this EMT, that is epithelial to mesenchymal transition, or sulfate determination, or tumorigenesis. That is, it will generate pro-tumorigenic properties. And the other things is the P13K, interact with this Muc-1C, it will promote this STAT13 pathways, that is PIP2 to PIP3 conversion, then AKT to mTOR, and thereafter that will lead to the upregulations of the proteins that are involved in protein synthesis, cell growth, or survivals. And this STAT1, STAT3, this is the different pathway, and this PIP2 and PIP3 are through this P13K. So this STAT1 or STAT3 when interact with this Muc-1 cytoplasmic domain, it can directly upregulates the uh, genes that are involved in angiogenesis, cell growth, or survival. Angiogenesis means providing some additional pathways for the blood vessels so that the food requirement for these tumor cells that is essential for the uncontrolled growth rate can be met through this angiogenesis process. And the last thing that is mentioned in this figure is about this Muc-1 cytoplasmic domain interact with this ERCX that will lead to the stabilized ERCX formation, and that will also promote the genes that are involved in cell growth and survivals. So there are different one pathways or one functions can be upregulated by different signaling pathways. The main point about this figure is that, so even if one can inhibit one kind of pathways, the other pathways can compensate or continue on its own way. That leads to the that will prevent the apoptosis or death of the tumor cells. So in order to counter this kind of signaling pathways, we need to act upon this deactivating this Muc-1 so that all these pathways will not get triggered, and the immune system uh, works on the immune system, or we can say the checkpoint system within the intercellulars of the cells works normally, such as this P53 works normally, and whenever it observes any unrepairable cell damage, it can trigger the apoptosis. So the best approach is to deal with this breast cancer is to somehow deactivate or prevent this Muc-1 from gets activated, and thereby this downstream Muc-1 cytoplasmic domain will not get activated. So this is the that main idea by which breast cancer can be easily cured. So I hope you got an idea how this Muc-1 plays a role in the breast cancer, and what should be the target strategy in order to cure breast cancer with the maximum efficiency. So this uh, approach is still under progress, and right now I don't have any information whether there are commercially available drugs available in whether there are commercially drugs available in the market that work on this strategy. You can search around the internet to check whether there is any FDA approved drugs available in the market or not. If you need to understand more about this role of Muc-1 in the breast cancer, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.